Please let me know if you cannot see my screen at any point of time so that I can reshare it accordingly. Okay, so hello, good evening. Welcome to the second session for the course on advanced concepts in cancer biology and precision medicine. As I've already introduced in the first class, my name is Ankita Bhattacharya. I'm a PhD student at IIT Kanpur, and I work on cancer biology as part of my thesis as well. So basically, what we have covered in the first class, I'm going to recap that quickly in a bit of a gist. We'll go through the slides just to brush up everybody's memory. But if you have any doubts at any point of time, you can go ahead and ask, or you can refer to the notes that are already being provided. So for starters, the first thing is that you must have gone through the flyer by now. So you have an idea of what to expect from this course. We have called it advanced concepts, but as you can expect, we'll be getting to that towards later stages as we progress through the course. Towards the last 3 or 4 sessions, they will be more in touch with what is being done in current research in today's time. The initial classes are more focused on what is known in literature, diverging away from what is known in the books for sure. But it will not be that intensive in the initial stages so that everybody can be, sort of caught up towards what we are eventually aiming to get to. So in the first session, these were the basic topics we had covered. We went through an introduction to cancer, the classification and different types of cancers based on where they are originating from. We understood some key terminologies for oncology, and we looked at the hallmarks of cancer. So basically, we first understood how cancer cells evolve, the main key difference being the fact that they are losing the loss of regulation on cell proliferation. And we understood the relevance or the contribution that has been made by these two scientists, Professor Douglas Hanahan and Professor Robert Weinberg. They have given rise, in fact, to the concept of hallmarks in terms of cancer. What exactly that is something you will obviously, you must have already learned it from the previous class, or it will be something you'll get to know in the upcoming slides. So we had understood the different substages that are present when cancer is progressing from a normal cell towards a cancerous state, starting from stages which involve some sort of alteration, which is usually, in most cases, it is a genetic mutation, how it progresses through hyperplasia, dysplasia, in situ carcinoma, invasive cancer, and finally becomes very lethal when the disease metastasizes. And we had discussed the different substages of metastasis as well. Following that, we had understood the definitions and looked at the pathological differences between different stages of cancer. So like you can see, tissue progresses from a normal state of organization through different substages involving hyperplasia, where cells divide a lot more rapidly, but the normal organization of the tissue is maintained or is kept intact. Through dysplasia, whereby cells become extremely abnormal, and finally they progress towards a stage which we define as cancer. Here were some other comparative images as well, and here you can see a gradation of tissue. Like within the same tissue, you can see different arrangements whereby it progresses from low-grade dysplasia, where the disease is much more mild, and furthermore, it progresses towards moderate and very high-grade dysplasia as well. If I'm going too fast, you can stop me. I'll try to explain that. Or of course, you can refer to the notes in your own free time as well. Following this, we had learned about the difference between benign and malignant disease. We had learned about the different types of tumors, which are classified based on what cell type the cancer has arisen from, starting off with carcinomas, which were the most abundant form of cancer that is most commonly diagnosed. We had understood the different types or different subclassifications within carcinoma as well. And before getting there, we had understood the difference between carcinoma in situ and disease that has become truly cancerous. So these were some of the initial stages that we had understood. Following that, we understood the subclassifications, starting off with adenocarcinoma, which arises from tissues which are glandular in nature, basal cell carcinoma, which occurs within cells of the basal layer of the tissue, squamous cell carcinoma, transitional cell carcinoma, all of them with different types of examples. When I say example, I mean the cancer types which are grouped under each of these categories. Then we had looked at sarcomas in a lot more detail. They are cancers that arise from bone and other soft tissues within the body. Some examples of the same were given here. All of the different subclassifications like Kaposi's, liposarcoma, etc. We had understood the difference between leukemia and lymphoma as well. I have mentioned that again in today's class, but to address a question that one of the students had asked in the previous session, so we'll come to that eventually. So leukemia basically refers to those cancers which are beginning in the blood-forming tissue of the bone marrow, and lymphoma arises from cells which are usually classified as lymphocytes. If you have studied immunology, you'll be familiar with them. They're basically the B and T cells of your body, the immune cells of your body, which provide you the major protection against all different kinds of pathogens. And the key difference between them was the fact that leukemia arises from bone marrow and lymphoma arises from the lymph fluid of the body. And we had understood some subclassifications of lymphoma, which includes Hodgkin's and non-Hodgkin's lymphoma. Then some other types of cancers included multiple myeloma, melanoma. Multiple myeloma begins from plasma cells of the blood. Melanoma begins from skin cells of any individual, or skin cancer is how you commonly know it. And of course, we had also addressed the fact that skin cancer is not very prevalent in our population because of the presence of melanin in our skin compared to the eastern cohorts, western cohorts, which are relatively a lot more pale, and therefore they lack melanin, and therefore they are more prone to these kind of diseases. And also about neuroectodermal germ cell tumors, and different tumors which arise within the spinal cord, within the central and peripheral nervous system as well. Then we had discussed hallmarks of cancer by Professor Douglas Hanahan and Professor Robert Weinberg, the 3 articles which have been published by them in the last 2 decades or so, 2.5 decades or so, and how they have established the different functional capabilities which are acquired by human cells when they progress from the state of normalcy to the state which you define as cancer. So under that, we had discussed the emerging, as well as we had just mentioned the emerging hallmarks and enabling characteristics, and I had asked people to go back and look into them, and we were supposed to discuss it today, but if it is not something you have read up till now, you can catch up over it, and we'll discuss it in our last class when we do a summary of the entire course. So there we had understood the key hallmarks of cancer. By hallmarks, we basically mean some characteristics of cancer cells which make them cancerous. So the first one of them was sustained proliferative signaling. This is something we will be getting to in quite a lot of detail today. Why so? Because today's theme, if I've not already mentioned it, is to understand the key signaling pathways in cancer. I have split it over 2 classes because signaling can get a little saturating to understand after a point of time. So today we'll we'll understand a few pathways today and a few more pathways on Thursday, so that you can learn it at your own pace, and they have been categorized based on some common characteristics. So we'll come to that eventually. But basically, what we understood here was the fact that sustained proliferative signaling, that is continuous signaling, which dictates that a cell should divide, that is a major hallmark of cancer and is a major disease-causing event. Following that, we had learned that evading growth suppressors, which means evading signals within the cell which prevent growth, that is another way in which cancer cells acquire this ability to become cancerous. The third hallmark we discussed was about resisting cell death, which included the different types of cell death, starting off with autophagy, necrosis, and the key mechanisms by which apoptosis is a major evasion of apoptosis is a major strategy by which cancer cells ensure that A, they keep on dividing, and B, they do not die by mechanisms by which a normal cell is supposed to die. So these were the first three hallmarks. Then we talked about enabling replicative immortality, which means that cells once they have started to divide, they keep on dividing and they become immortal. And that system is switched on by cancer cells. And we understood the key mechanism for it being the fact that telomerase, an enzyme, is overexpressed, which allows this to happen. Then we understood that inducing angiogenesis, which means the synthesis of blood vessels within cancer cells or around tumor cells, is a major strategy by which they keep on supplying the tumor with enough oxygen and enough nutrients so that it can keep on dividing. Because if you have studied cell division, you would know it's a very energy intensive process. So a lot of resource goes into allowing a cell to divide from 1 to 2. So if cancer cells need to keep on dividing, they need the energy stock to be able to do the same. And that is facilitated by an angiogenic switch, which is basically the activation of multiple pathways that switches on the system that allows cancer cells to keep on sprouting new vessels. I had used this terminology in the previous class. You can go back to the recording for your reference. And then they form these abnormal, haphazard, they form this network of vessels, blood vessels, which allows the supply of nutrients and oxygen into the cell. And finally, the last hallmark that we discussed was activating invasion and metastasis, which means that, okay, now cancer cells have, become this huge mass, they have this blood supply, and therefore they are, happy in their own location. But for the disease to become truly, to reach its full potential, it has to move to all parts of the body, and it does so by the process known as metastasis. I had also asked everybody to just go back and quickly look at the different substages of metastasis. I'm not sure if anybody has done it. If not, that's again something we can address in our last class for the entire course. And under this hallmark, we had discussed this major process known as EMT, which is epithelial to mesenchymal transition, which is a cellular program by which epithelial cells, which have a polarity, which are normally present as part of some tissue or part of some organ, they become mesenchymal in nature. So their ability to hold on to a layer that is lost, and they become more like it is more feasible for them to not stay bound to a location, but instead move throughout the body, which allows them to spread a lot better and cause a more serious disease in other parts of the body as well. So a tumor which has, say, for example, started, like I discussed, I work on prostate cancer. So a tumor which has started in the prostate gland can eventually metastasize to other parts. The most common site where it metastasizes is the bone. But apart from that, it also metastasizes very often to the lung, to the livers, and to the brain. These are the three very common sites, but bone is where prostate cancer metastasizes the most frequently. Again, under this domain itself, I had posed one more question, which I think is very, very important. And I had discussed it in the context that it might be very relevant if you eventually want to appear for some interview in the long run, which is the seed and soil hypothesis. If it is something you've already studied, very good. If not, let me know. That is something you should definitely learn about. It basically is the idea, if I just sum it up very quickly, it is the idea that any cancer or any cell for that matter, in this context, we're talking about cancer cells, they cannot, like for example, think of it as any crop. Not every type of crop can grow very well in every possible soil. You can't grow rice, right in your backyard. You can't grow every type of crop everywhere. So there is a concept which is the seed and soil. So the seed which you are sowing and the soil on which you are sowing it or in which you are sowing it has to be compatible. So this is the logic that is used by farmers, and that is the logic which was translated by a scientist known as Steve Paget. And Paget had explained that much like this concept, very similar to that, cancer cells also cannot just grow anywhere and everywhere. Initially, they cannot. What was instead proposed by him was the fact that there are some sites within the body which provide an environment that is a lot more suited for cancer cells to grow. That environment if you use the correct jargon, the correct scientific terminology, that is known as the microenvironment. So the tumor microenvironment is basically what dictates whether or not a tumor can form there. So in most cases, if you have heard about people who, who have suffered from cancer, if they, God forbid, if they progress towards advanced stages, most people, more often than not, have cancer that has spread towards the bones. That is because the bones provide a very good site for metastasis. But not every cancer metastasizes to the bone. Not every cancer metastasizes to the lungs or to the liver. Again, read about this as well. Have you ever, not have you ever, have you heard about the frequency at which people suffer from cardiac cancer, from heart cancer? It happens. It's actually cancer of the cardiac muscle that sometimes happens, but the frequency at which it happens is very, very, very low. You will barely find any reports. One of the major reasons for that is the fact that the heart is a tissue that is continuously having some sort of motion, right? It is continuously pumping blood. So for that reason, tumor cells will never get a stable environment where they will not face a mechanical pressure by which they can move out. There are other reasons for it as well, but this was just the most obvious reason that was, I think it has been discussed in one of the hallmark papers as well. I'm forgetting which one, most probably the first paper itself. But yeah, that is where it was discussed. So compared to that, a tissue which does not have any blood flow, any fluid flow, that will be a lot better, a lot more stable for a cancer cell to localize. And that's why bones are an excellent place for them to grow. And as stable as it is, the more difficult it is to eliminate cancer from the system or from a person's body who has progressed to that stage. So win-win for cancer, but the patient obviously is more often than not able to actually make it through the disease. So read about this in a little more detail about seed and soil hypothesis. This I just I verbally explained it very quickly. You'll find fantastic notes about it. If you don't get it, if you want more references, you can drop me an email or you can, mention it one of the classes. We can have a quick discussion in just 5 minutes about it. So we had summed up the entire class. I'm not reading it again because it's just what I discussed. But yeah, I had asked everybody to go through these few topics, read about autophagy, its pro-tumorigenic and anti-tumorigenic roles. I have not included it in today's slides because I want people to actually go back and read about it. Because if you think about any process, like a cell death process, then how can it be pro-tumorigenic? It is supposed to help the cell survive or maintain homeostasis. So it is a question that should be read about by students themselves. But of course, I am there to help you out eventually in the long run. But give it a shot, try reading about it. It is interesting. The different substages of metastasis were, if you remember the first slide where I had shown the progression of cancer, where the substages were mentioned. So I wanted everybody to go and look at it once. If not, I'll explain it. It'll just take like 2 minutes. I had already explained it briefly in the previous class. But yeah, there are different substeps, which starts off with dissemination. There is intravasation, extravasation, and then again, the localization of the tumor at the new tissue. I had also asked people to go back and look at emerging hallmarks and enabling characteristics and understand their difference from core hallmarks. This was directly supposed to be studied from the third hallmarks issue, which has been published in 2021. So you can go back and look at it as well. So if there are any queries till now about what I've just discussed, you can just unmute yourself and let me know. I'll try to clarify that. So please don't hesitate. Any question, whether you feel confident about it or not, please go ahead and ask. I'm also a student. I'm also learning, much like you. So any queries, please let me know, and then we can start today's session. Okay, if not, if there are queries during the course as well, you can just unmute and go ahead. We'll discuss it then. Don't worry. Okay, so coming to session 2, uh, like I have previously mentioned, today's class will look at key signaling pathways in cancer. We'll start off with one of the unanswered questions. Then we'll understand the components of any signaling cascade. We'll learn about some types and classifications of signaling pathways in cancer. oncogenic pathways as therapeutic targets. So again, this is a topic that will be covered in today's class and the next month. So some part of it might not be complete today, but that's the overall goal. So look at both of these classes as one consolidated unit. Um, what the video or GIF, whatever, what you see here is something you might be familiar with, but that is something we'll discuss eventually. Just for starters, just look at the fact that there is something which is sitting on the, there's this sort of an orange moiety which is sitting on the receptor, and then there's some lots of things that are happening downstream of it, which is basically what forms a signaling cascade or a signaling pathway. Excuse me. Just a second. Okay, so something that was discussed in the previous class and was asked as a query by one of the students was about lymphoma and leukemia. So the question was that, is lymphoma a part of leukemia or vice versa, or what exactly is the situation? So very clearly, this I took just off Google Images, but what I had mentioned in the class is basically the correct answer, and that is what I'm just reiterating here. So leukemia and lymphoma, very commonly, they are known as blood cancer or liquid cancer. They do have a common origin. The only point of overlap between these two types of cancers is the fact that it arises from the immune cells or the white blood cells of the immune system, which are lymphocytes, which includes lymphocytes as well. However, they are not the same. They cannot be grouped under the same like one is not a subtype of the other. The main umbrella term remains blood cancer or hematological cancers, but under these, there are different subclassifications. You cannot group them. Leukemia remains the cancer of blood cells that starts or that begins in the bone marrow and then it travels through the bloodstream. However, lymphoma is cancer of the lymphatic system that usually originates in the lymph nodes or sometimes the spleen. Just for reference, this is the difference between the two. Both of them can eventually move into the circulatory system because that is usually the route that is chosen by cancer cells for metastasis. It's the most convenient way for them to move about. And however, the site from which it has originated remains different, and that is the classification difference between the two. So I hope that is clear. So that was a question that I wanted to address. Now, moving ahead. So we are going to quickly go through the different components of a signaling cascade. You might have studied it already, but because people are coming from different different levels of study till now. Some of you are postgraduate, some of you might be undergraduates or might not have just studied it in general. So just to brush it up. So basically what you need to understand is that there's a difference between these terms as well. I have used cascade in most places. The more accurate terminology would be signaling pathway. A cascade usually is used for a process where there are multiple where there's a default, sort of a default setting that protein 1, protein 2, protein 3, protein 4, they're all aligned. As soon as protein 1 gets the signal, the default pathway will be followed and everything that happens right after it forms a cascade. Signaling pathway is a little more relaxed when you see it as a terminology. It includes any pathway and there from point 1, from protein 1, protein 2, protein 3 or protein 4 might remain as options instead of just being the most default route that is to be followed. So sometimes they're used interchangeably. That is how I've utilized it as well. But I just want you to learn the difference between the two so that you are more mindful about using it. Cascade, the terminology cascade is more commonly used for the pathway that is followed in apoptosis. I will be discussing it in the next class. So next class, we will look at apoptosis. We'll look a little bit at autophagy and we'll look at some of the steroid hormone pathways because they're very, very relevant to cancer and cancer progression. They are key oncogenic events in cancer. We'll learn about that. But for now, what we want to understand is what exactly comprises or what exactly forms a signaling pathway or a signaling cascade. So there are a few components. Think of them as coordinators of a project. So first of all, this is a model of a hypothetical signaling pathway. example being, this is one that operates usually in yeast. Why yeast? Yeast is a very commonly chosen and has been a very commonly chosen system of study for most eukaryotic processes before people had understood Drosophila, people had understood mice and all of these, because they are more complicated organisms and there's not a lot you can manipulate or modify in these organisms, and there are a lot of ethical questions also that arise there. In contrast, if you think about yeast in general, that is a lot easier to use as a study model. So in this hypothetical signaling pathway, you are looking at a few things. First of all, this is your cell membrane. This is the extracellular side outside of the cell. This is the inside of the cell. Now, there are some intracellular signaling molecules shown here in green, and there are some target proteins which are beneath it, which are shown here in blue. So what exactly is happening there is that under normal circumstances, when the pathway is not switched on, nothing is happening. They're all in their own place. When you're switching this circuitry on, there is an external or extracellular signaling molecule that comes and that sits on the receptor on the surface. Then you can see there's an alignment of protein 1, protein 2, and protein 3. Intracellular signaling molecules, which were just roaming about, have now come into action, and now they are all responsible for completing this pathway, which has been initiated because of an external stimulus. So what now happens is 1, 2, and 3 are doing their job. 1 is acting on 2, 2 is acting on 3, and then 3 is executing whatever downstream activities are supposed to happen as a result of that pathway working via these downstream target proteins. So these are also terminologies I hope you're familiar with. Upstream basically means, if you think of a process, whatever comes in the beginning or towards the initial stages is upstream, and whatever comes towards the end is downstream. However, think of them as sort of relative terms, like for example, like how you would use greater than, lesser than, much like that. So if there are 10 proteins that are working in a particular pathway, then protein 5 is upstream of protein 6 and protein 8 is downstream of protein 7. So it's just relative, however they're aligned one after the other. So let's read the legend. An extracellular signaling molecule has bound to a receptor. Usually they span the plasma membrane and has activated a series of intracellular signaling molecules which activate these target molecules and affect changes in some processes. Here they have shown, just as an example, altered metabolism, altered cytoskeleton, and altered gene expression as well as cell division. So this is the basic architecture of any signaling pathway. What you need to keep in mind is that the most default setting has been shown here, where you have a receptor. As the name receptor suggests, it is the protein, or it is the entity that receives the signal. What is downstream of it, after it, or beneath it? They are the transducers, which means that they are transducing. They are listening to the signal. Think of it as somebody who is coordinating a project once again. So there's a manager who is distributing some sort of whatever instructions they want to deliver. The person who is sitting in charge of another team is taking their commands into consideration and is then giving out the commands that need to be executed by those who are somewhere at the bottom of the entire hierarchy. So that's pretty much how it happens. There are some things which are just transducing the signal. They're not doing anything else. And there are some proteins which are executing the activity that needs to be done. So more often than not, if they are proteins, which this is actually among the most abundant proteins within the cell, if these proteins are able to move into the nucleus and they're able to bind DNA, and then they're able to cause changes in in transcription, etc., then they are transcription factors. In most cases, these are the effectors of any signaling pathway. However, apart from this most default setting that we have discussed so extensively, there are other types of receptors as well, as you can see here. So as you can see here, and they are under this schematic, you're only looking at membrane receptors. There are other types of receptors as well, one of which we'll discuss in Thursday's class, which are steroid hormone receptors. So here you can see that there are ion channel receptors. Basically, they are dependent upon the binding of ions, and usually there is also a coactivator that also binds along with it. And the binding of the ion or the influx of the ion is what is actually working as a receptor, and then it does its downstream job. Then this entire group of proteins, which are known as GPCRs, G protein coupled receptors, again, a whole field of study entirely by itself, a very, very important component of cancer signaling. Maybe we can discuss it very briefly in in Thursday's class, but they are also known as 7-pass or 7-helix transmembrane receptors as well, because they pass the membrane 7 times. So if this is the membrane and this is the receptor, so 1, 2, 3, 4, 5, 6, 7. So they're going across 7 times. Then there can be a receptor that has intrinsic enzymatic activity, which means that something is binding at the top, and the receptor itself is an enzyme which is able to initiate something downstream. It does not need any transducers. It is capable of acting by itself. And then you have an enzyme associated receptor, which is a recruiter receptor. So once the enzyme, which has been shown here as a green protein, which are intracellular proteins with enzymatic activity, they are co-localizing, or they are sort of scaffolding onto this. Scaffolding is basically like a support. They're using the receptor protein as a support, and they're binding onto it on the surface. So when the ligand is binding to the receptor, then it is leading to the activation of these proteins. So we'll come to this fact. Why are we concerned about cell signaling for cancer cells? But before that, do you have any doubts, any questions?
No, ma'am.
No? Okay, so let's discuss this very superficially. Why are we worried about the fact that cell signaling is affected? I had discussed this during the hallmark session in the last class as well, but if somebody else wants to give some inputs, so we are talking about the fact that cell signaling, any cell, normal cell, cancer cell, whatever, has a signaling system that is working within it. So why are we worried about it? It works and it is fine, it is helping cells survive. So what is your idea about it? That what is going wrong? What is the problem? Why are we concerned about it? Any inputs? It may be right, may be wrong. Don't worry about it. Any inputs, please share.
Ma'am, because the cell signaling is important for cell growth. Like the cell signaling regulates the cell division and growth. So
Mhm.
maybe if some kind of mutation happens and the cell signaling, the pathway doesn't work correctly, then it may lead to cancer. That's why we are focusing on cell signaling.
So, what do you mean that the pathway stops or the pathway is not working? It is partially working, it is overworking. What is possible? What could be a cancerous event?
It can cause overexpression of some protein, or maybe it can also stop completely.
Mhm. Right. So you're correct. So these are obviously the most possible ways in which cancer can actually arise. That the pathway which is supposed to be functional, that is stopping. Um, but let's address this question a little from a bird's eye view. Suppose there is a pathway X, which is very necessary for the cells to survive. Think about it in this way: any process that occurs within any normal cell under normal circumstances is a pathway that is necessary for the cell to survive. Why? Because the cell does not have the luxury of having excessive resources just in stock. Whatever resources a cell normally has will always be utilized in the most proficient, most productive way. So if a pathway is switched on under normal circumstances, it is switched on because it is necessary to switch it on. So if within a cell, this pathway X is shut down, then what is going to happen? Shouldn't a cell die instead of becoming cancerous? That is one possible question that needs to be addressed. Secondly, if a pathway is overexpressed, why is it a problem? It should, actually aid better survival of cells. So there are a lot of ifs and buts when we talk about cell signaling for cancer cell. And apart from the fact what you just mentioned, I think it was Ankit. Yeah. So what you mentioned, you are correct in a lot of ways, but there are a lot of nitty-gritties that need to be cleared out about it as well. Why so? For any signaling pathway that is operating in a normal cell, there are a lot of feedback loops that operate to maintain cellular homeostasis. Even within a normal cell, any pathway cannot be switched on indefinitely. There are a lot of checkpoints, checkposts. I think I might have mentioned this in the first class as well, whose job is to ensure that the pathway is switched off as well at the right time. Like I said already, that production of energy for a cell, if you have studied metabolism, if you've studied the entire electron transport chain, like the entire process of glycolysis, etc., and ATP generation, that's a huge process. There's a lot of energy, a lot of ATP that goes into producing ATP. So therefore, cell is very careful with the usage of resources. Therefore, a lot of checkpoints have been put into place. What sort of checkpoints are these? There are some feedback loops. What does it mean? The term feedback loop basically means that the cell is receptive to feedback. The cell is sensing what is happening within it and is responding, in accordance with whatever feedback it is getting. So for example, we'll take each of them one by one. There are three major types of feedback loops that are present. The first one is a feed forward loop. The second one is a positive feedback loop. And the third one is a negative feedback. So for the first one, feed forward loop, what do you see here? You see protein A, B, C, and something called Z. And you see that for all the others as well. What is the meaning of this? This means these proteins, A, B, C, and so on, they are all part of some signaling pathway, let's say X. But what is crucial to understand here is the side arrow. Do you see each of them is different for each type of feedback loop? For the first one, you are seeing that the arrow directs from A to Z. For the second one, it goes from Z to B or A, depending on where it is. And the negative one shows a different type of arrow. So if you're not already familiar with it, the arrow basically means it is an activating step. Activating as in it will switch that protein on or switch that pathway on. And this sort of dash or this sort of line is an indicator of an inhibitory protein, which means the process stops. It doesn't go ahead. So what do these three types of feedback loops exactly mean? A feed-forward loop, or as I've mentioned here, an FFL, shown in the figure here, is a network substructure. Here the reference has been given differently as if X, Y, and Z. Let's take A, B, and C in this case. So you have A, B, C. All of them are connected to Z. This is the term loop in this case is a misnomer. Instead, what it has two possible pathways. One, where you have A regulates B, B regulates C, C regulates Z. So this is the direct regulation. The other possibility or the other um, way in which this loop or this pathway is fed better is by the fact that A can also directly regulate Z as well, bypassing all of these. So what basically happens here is that think of it as a process where some signal X has come in and it has switched on the first protein A. Then what can happen? If why is this necessary? We'll come to that later. Then what will happen is that by the most default route, A will activate B, B will activate C, and finally Z is getting activated. However, why is this arrow necessary? Why is this extra feed for taking the step forward necessary? That is because in some processes, this is most commonly seen in bacteria like not yeast, sorry, in bacteria like E. coli, sometimes also seen in yeast as well. So what exactly is the necessity there is that depending upon some external stimulus, there needs to be a very sudden and very huge increase in the expression of some proteins. Now, keep in mind that these are mostly prokaryotic organisms, so their system or the the resources or the intracellular system that they have for the overexpression or the increased expression of any protein is relatively weaker than what we have, right? So in that case, what happens is that you need to have a lot of uh, activating signals or a lot of positive signals to increase that expression very suddenly to a very huge amount. So therefore, instead of just depending on one pathway, a default pathway, just switching it on helps in direct activation and helps in increasing the cumulative signal which will be recognized by the cell. What also you need to keep in mind here is that more commonly these are transcription factors itself. So the activation of the transcription factor happens a lot quickly, and therefore the transcription or the expression or whatever is the downstream effect of this feed-forward loop, that is taken care of a lot faster. Now think about this. Having just this loop will actually do more harm than good for a normal cell that is showing some signs of becoming precancerous. That is because instead of just keeping a check on the fact that this is a necessary process, it is just keeping on switching that process on without any further check. A similar sort of process can be seen with positive feedback loops as well. So positive feedback, as you can understand by the name itself, it's here I've shown the most commercial definition of this particular terminology. Positive feedback basically is a self-perpetuating pattern of behavior where the end result is reinforced by the initial act. So it's a loop. This is a loop. How so? Switching on protein A activates protein B, C, D, so on and so forth, and finally protein Z. The more protein A is switched on, the more protein Z will be made. And because this is a positive feedback loop, the more protein Z is switched on, the more protein B and protein A will be made. So basically, it is a self-sustaining loop whereby production of A increases production of B and B helps in increase production of A. So it's a continuous infinity loop whereby the feedback is going on in a positive manner. So again, this is what it basically means, that the end result is reinforced by the initial act. So more amount of end result leads to more initial stimulus and so on and so forth. In exact contrast to both of these loops, whereby feed forward and positive feedback, both of them are operational, in exact contrast to that, the negative feedback loop works. How so? So the negative feedback loop is the most necessary. It's a very finely regulated but a very crucial procedure or a very crucial response that is usually found in any living organism, whereby the after effect or the need for that is to stop a reaction or to slow down that reaction as and when necessary. So once a process A has been initiated, or once a protein A has been activated and the downstream process is initiated, once the final product Z is produced, more the amount of protein Z is produced, more it will tell the cell that, okay, enough of me, I don't need to be produced any further. So shut down that process. So it is giving feedback to the upstream coordinators or the upstream transducers that, okay, this process now needs to be shut down. So these are the majorly three types of feedback loops that are very crucial, that are operational in almost every living organism, and that are necessary to maintain cellular homeostasis. Keep in mind that this is not a necessary, it's not always important that every pathway within the cell needs to have a feedback loop. No, in most cases, actually it does not happen. But in processes which are very finely regulated by the cell, like I explained, something which is very energy intensive has a lot of checkpoint steps. So if you have read glycolysis, this is something you will understand that the investment of energy at different steps is different. And any step that has more expense of energy or more investment of energy is considered to be a rate limiting or a committing step, basically, that now the process will go forward. And if that step has not been completed, then the procedure will be stopped. So basically, these are the three types of ideas that I wanted you to understand. Any doubts, please go ahead and let me know. You might be familiar with it, but if not, just let me know. And then we can have a discussion on it. Okay, if not, then coming to the fact, then what goes wrong in cancer? Like Ankit had already mentioned, a major problem that we all are pretty much familiar with is the fact that there are mutations in cancer pathway genes. Secondly, this is something you might not be very familiar with, but basically the tumor microenvironment that I mentioned during the recap session, there are a lot of epigenetic changes that occur within the tumor microenvironment. How and what, this is a very fairly advanced concept, especially if you're not familiar with the TME, the tumor microenvironment. But basically, depending upon the type of environment that the cancer cell is exposed to, a lot of changes can occur within the genome of cancer cells. When I say within the genome, I don't mean in the genome sequence. I hope you understand these terminologies, what genome exactly means. Genome is basically the entire genetic material, the DNA of the cell. So one way in which changes can occur are by mutations in pathway genes. Now, mutation means that within the sequence of DNA, within ATGC, there is a change, as a result of which A is no longer A, T is not T, and so on and so forth. So this is within the sequence of DNA. When we say epigenetic changes, that is not a change that has occurred within the sequence, but instead it is a change that has occurred on the sequence. When I say on, it basically means modifications like most common modifications being methylation, which is the addition or removal of methyl group, CH3 group, or very commonly also seen acetylation and deacetylation. So if you have studied epigenetics, you would be familiar with these concepts. So there's demethylase, there are demethylases, there are deacetylases, there are histone demethylases, acetylases. All of these are occurring. But for DNA, the most common uh, change that occurs is methylation, especially in the CG islands, CPG islands present within the genome. So these changes can, while it would normally not be present, some change that might not be normally present, that can occur in a favorable tumor microenvironment that actually aids and that influences the development of cancer. Thirdly, there can be metabolic reprogramming. This is a whole other field, learning about cancer metabolism. I'm not going there. I'm not even an expert in that field. We can very briefly discuss it because it will be part of the signaling pathways that we will be discussing. But metabolic reprogramming is a huge, huge change that does occur in cancer. It's something that is very interesting, very fleeting. It happens very quickly, but yeah, it has a huge role to play. And finally, the last point, which is something you all might be familiar with, and which is the crux of our entire class as well, which is the loss of regulatory control on all of these signaling pathways. So signaling pathways are there, fine, all good. But then any loss in the control, in the regulation or the coordination of these pathways is what eventually leads to cancer. So with this pretty long introduction in mind, we will come to some, I'll touch upon, the idea of how signaling pathways are changed or altered in cancer cells. How do scientists actually study that? So if I'm not sure if you guys have some, studied from any papers, if you're familiar with it, if you explore PubMed as part of your current studies or not, but I just wanted to familiarize with you. So basically, we now know the fact that cancer signaling pathways are something which is behaving abnormally. There is some problem in the normal signaling pathway. The question is, how would you ever figure it out? So I found this very, very beautiful paper. You can refer to it. It might be a little advanced for somebody who has not had background, but you can just go through the images. Just go through it. It might intrigue you, maybe not. It's something that can trigger an interest in you eventually. So basically, what these scientists have done, this research group by Sanchez Vega, they published this article in Cell in 2018. So they have studied oncogenic, their objective was to study oncogenic signaling pathways in the Cancer Genome Atlas. Now, what is this Cancer Genome Atlas? The reason I'm putting all of this is because these are, if you want to go into cancer research in the future, you learn these eventually. The earlier you learn, the better, the more ahead you would be of the rest of your peers, basically. So basically, across the world, research groups, what they have done is there are different attempts. Okay, let me put it a different way. Think of it in this way, that people across the world are trying to figure out cancer and cure cancer. How do they do that? They basically submit the data of cancer patients from their own countries in very different ways into common repositories. These repositories are used by scientists across the world, and people study it and people figure out, okay, these XYZ things are wrong and these XYZ things can be targeted, and therefore we can figure out a way to tackle this disease and help people. The end goal is helping people. And in academia, it's because people love working on cancer. The disease is fascinating, so people stay there. So basically, this Cancer Genome Atlas is one such repository. It was a huge initiative by scientists from across the world where they sequenced the genomes of patients, and all that data is publicly available. You can access it. I do access it. You can access it from CBio portal. This is a bioinformatic tool. Again, yeah, this is something you also, if you want to work in cancer, if you want to work, like you want to do a PhD or you want to become a researcher, um, these are resources you will get familiar with over time. But for now, what we're just trying to understand the way in which people work. So this repository is available online, and it has the sample of patients with many different types of cancers. In this paper, the scientists that we are mentioning here, Sanchez Vega and their group, they took the sample information or the tumor information from 33 different cancer types. So this, as you can see, these are the different types of, these are some of the examples of the different types of cancers. I think I mentioned it here. Yeah. So again, this I put all of this. Don't get scared by it. It's not something you need to, it's not for rote learning. It's just to pique your interest and to get you a little in touch with the way terminologies are used everywhere. So these are all of the different types of cancers that they took. And under that, you can see that the total number of tumor samples basically means the patients from whom the sample had been taken, and that has been sequenced and that has been studied. Again, sequencing is a topic we'll very briefly cover in, I think, the second last or third last session, because it's not something that I am particularly going to focus on in this course. But yeah, you should be abreast with what is going on in the world. So basically, what I wanted to show here was that these 9125 tumors, they all have these different sources of origin: breast tumors, thoracic, endocrine, head and neck, eye, CNS, either hematological or lymph, soft tissue, skin, gynecologic, urologic, developmental, GI tract, core GI tract, and that is the broad classification. Within each of these, you see these strange forward terminologies. They are basically TCGA terms, which are for each of these disease codes. You can find them completely explained here. But basically, these are all of the different subtypes. Like you have AML, acute myeloid leukemia, you have PRAD, which is for prostate adenocarcinoma. That is what I work on. You have breast cancers, you have colorectal cancers, you have ovarian cancer, etc. So the grouping is very different. You don't have to worry about that, but just getting you an idea, just giving you an idea about all of this. So they took all of these different types of samples. They studied the mutations, the copy number, methylation, like I just explained to you, the expression, and fusions. I hope you understand what copy number means. So in cancer cells, one more change that occurs very frequently is gene duplication or gene amplification is very prevalent. So basically, any gene, any oncogene that is very dangerous for us, for a normal cell, if it starts becoming beneficial for a cancer cell, what it will do is it will just increase it in number. It will be due to malfunctioning or improperly functioning DNA polymerases that are responsible for DNA duplication or DNA replication. So instead of there being one copy of that gene, there will be tandem copies. Tandem means back to back. So suppose you have, let me give you an example of a gene that I work on, which is AR, that is androgen receptor, which is a steroid hormone receptor within the cell. So within a prostate cancer cell, what happens? One of the things that happens is that the AR gene is amplified, or it, instead of there being one copy on one chromosome as one allele, there are, say, 50 copies of that gene. As a result, more the number of copies, more the amount of transcription, translation, more the amount of AR, more amount of disease, the patient dies. Again, a very it was quite rapid, but that is one of the things that happens, and it is a major challenge that is being tackled for the disease. This was just one example. For every type of cancer, you'll find some or the other change that occurs, and copy number change or copy number amplification, CNA, as it is normally understood, is very, very common. And apart from that, your methylation and expression, etc. All of these different things were studied. What they have done in the paper is that they have identified the 10 most commonly altered pathways. I've not mentioned it here, but yeah, the different types of pathways that are most commonly uh, prevalent in cancer patients and that are considered to be cancer causing. And finally, they have also elucidated about the different types of drugs, more than 100 plus drugs. This includes those that have been approved, that are in clinical trials, that have been, that has some potential resistance associated with it, all of it. So that's just the idea for you to keep in mind. Again, I am not going into this paper. I just wanted to get you a little bit interested. I hope it has gotten you interested. But yeah, that's how people today, till date, are looking at different types of pathways. We are taking patient data um, and we are trying to figure out what went wrong. So it's like a bottom-up approach, basically. You start with the sample, you go back to the start of the disease. And the top-down approach is where you know the beginning of the disease and then you go towards the progression or the end result. So, okay, let's move forward. I'm going a little slow, but yeah, we will be able to figure it out, don't worry. So the first pathway that I want to discuss today is known as the RTK RAS signaling pathway. Does anybody know what is RTK? Have you studied it? Please unmute and go ahead if you know about it.
Receptor tyrosine kinase.
Correct. Uh, Anybody else? Somebody else, please also interact. Have you studied it? If not, then I'll just quickly, you will understand it from the slides, but okay, let me just move ahead. We are running a little slow. So basically, like he mentioned, RTKs are receptor tyrosine kinases. They are a family of receptors which act as enzymes as well. If you remember that first diagram, not first, but that diagram where I showed that the receptor itself has some enzymatic activity, that is actually what RTKs are usually classified under. So RTK, it has a lot of possible downstream effects. We'll come to that. But one of the major downstream effects that is mediated by RAS is often an oncogenic event. So we'll come to that first of all. So how does this RTK exactly work? Basically, this receptor tyrosine kinases, they are usually almost in all cases, there is one exception to it. You can read about it. These are almost always transmembrane receptors. Transmembrane basically means it spans on both sides of the membrane. Keep in mind these terminologies all mean different things. Transmembrane means it has to pass across the membrane. So this is your membrane, there's an extracellular and there's an intracellular side. In some cases, there are membrane embedded receptors, which means that instead of there being this extracellular side, the receptor goes like this and it comes back. So it is only present, the extension of that receptor is only present on one side. They are a different class, but RTKs are mostly transmembrane receptors. Here you see that the receptor has its most characteristic feature is the fact that it has a tyrosine kinase domain. What is tyrosine kinase? Kinase, if you know the function of it, is basically any enzyme that is able to add a phosphate group, PO43 minus, onto whatever is its target. This process is known as phosphorylation, right? So when there is phosphorylation by RTKs, when there's phosphorylation at tyrosine residues, that is mediated by RTKs. How do they work? So here you see this is an inactive receptor where the two components or the two monomers of the receptor tyrosine kinase are separate from each other. When there is binding of the signal molecule, in this case, this particular example is talking about a signaling molecule or a ligand, which is a dimer in nature. So after dimerization, it binds to the receptor's outer or extracellular side. And upon binding, the two receptor monomers, they come close to each other and there is interaction between the two tyrosine kinase domain. So this kinase activity is stimulated through this dimerized RTK. After this dimerization has occurred, this activity is stimulated and there is phosphorylation of tyrosine residues within the receptor itself. This step is called as autophosphorylation. Autophosphorylation means self-phosphorylation. When it is phosphorylating something else, then or if it is phosphorylating different parts of itself, then it is called as trans-autophosphorylation. However, in this case, there is autophosphorylation, and then what happens? Signal is relayed by the activated signaling proteins into the cell's interior. So phosphorylation of this particular receptor allows the docking or allows the binding of all of these different proteins. You see here green and sort of yellowish and purple, all these proteins are now able to bind once that place has been phosphorylated. And this binding is relaying some signal which is downstream of it into the cell's interior to control some other pathway. This is how it overall, this is the way it works. Here, what I've shown, this has actually been taken from the paper that I just discussed quite extensively about TCGA, and they have just given examples. You don't need to stress about it, but they have just given examples of all of the different components of the RTK-RAS pathway that they found mutated in the paper. I would very strongly urge you to go back and study it. There is a code for this as well. So the color that you see for each block, for each protein component, that is indicative of something. I want you to go back and read it. The more red it is, the more blue it is, it has some implications. Please go back and read about it. It'll be very interesting and it'll help you understand how to read papers. Basically, you can just look at this diagram and you can draw like 20 conclusions from it. So that is something you need to learn over time. But basically, overall, what you see here are RTKs or receptor tyrosine kinase. These are all the different names that have been suggested for different types of receptors. They are all RTKs. It's a family of receptors, but these are all having different types of downstream activities. Then you have RAS, to which RTK signal is being carried forward, and then the downstream effect is taken care of by some other proteins, ultimately, which helps in controlling cell proliferation, cell survival, and translation. Just excuse me a moment. Okay. So that is what happens in the default RTK-RAS signaling pathway. The question is, how does it play an oncogenic role? We'll come to that very soon. But before that, let us look at the pathway in slightly more detail. Just a sec. Yeah. So this is your inactive monomers or two inactive monomers which are present under normal circumstances. This is actually a better diagram for understanding as well. You see this is a ligand, accessory molecules, extracellular domain, transmembrane, juxtamembrane, tyrosine kinase, cytoplasmic domain, and phosphate group. So these are the different forms in which the active receptor can look, different ways in which you can visualize the active receptor to which the ligand has bound. And what you see here indicated as the cross shows trans-autophosphorylation. Now, once phosphorylation has occurred, what exactly happens? Just slightly, I'll read about it a little bit. So, mechanisms in order. This is a ligand mediated, receptor mediated, or mediated by a combination of ligand and receptor with or without accessory molecules, all of which leads to trans-autophosphorylation and activation of downstream signaling network. Various RTKs also can have asymmetric dimerization. Here only symmetric dimerization has been shown. Then what happens downstream of it? Once this phosphorylation has been completed, you can see there are a lot of different types of proteins. Don't worry about it. We'll come to it eventually. But you can see that it leads to the docking of multiple proteins. For example, GRAP2, SOS, RAS, or it can initiate another pathway via PI3K. We'll come to that next. There's PLC gamma, which is another way in which initiation can take place. All of these pathways are leading to control over cell survival and metabolism, cell migration, proliferation, differentiation, cell cycle, and control. What I also want you to see is that you see some random proteins again in the middle. They are also carrying this yellow colored phosphate group. So Who phosphorylates them? How is that happening? All of these are questions you'll also, you'll learn it eventually. But just look at it from a bird's eye view. The basic steps are there is receptor monomers which are present. There is ligand binding, receptor dimerization occurs. Phosphate tyrosine receptor tyrosine kinases are activated. They are causing trans-autophosphorylation. They're allowing new proteins to come in, and then cell survival and metabolism is being maintained in a normal cell. This is a necessary pathway which helps in maintaining normal cell survival, cell proliferation, etc. Now, there are four well-elucidated signaling networks, like I just mentioned. One, which is the RAS, MAP kinase pathway. You have the PI3K AKT pathway. You have the STAT3 mediated pathway and the PLC gamma protein kinase C pathway, all of which are responsible for controlling or for uh, regulating a myriad of biological outcomes, the outcomes being all of these. So this is what happens in a normal cell. Now, what I also want you to understand is that this response can be against very different types of proteins as well. So as you can see here, this is a schematic representation of the MAP kinase pathway and the various crosstalk signals that can also operate here. So you have let me reframe it. Basically, what I want you to understand is that there can be different types of signals, all of which can eventually merge towards the same downstream pathway to have the similar sort of downstream effect. For example, you can have growth factors, you can have survival factors, you can have chemokines, you can have death factors and ECM, all of which are eventually coming into contact or cross-talking with the same pathway, which is the RAS-RAF mediated pathway. And what it eventually affects are the same processes like we discussed already: cell proliferation, survival, differentiation, motility, and angiogenesis. So this I just wanted to bring to your perspective. Now let us understand how the oncogenic role of this particular pathway is coming about. So like for example, I think we have already briefly discussed this, that there can be a overexpression of our receptor itself. Very common thing. Like I just mentioned, that if there is an amplification of the AR gene, for example, again, keep in mind AR or androgen receptor, like I mentioned, it's not a cell surface receptor normally. But if there is a cell surface receptor, you have more people basically looking out. You have more security guards at your door. So more number of people that can recognize that, oh, there's a thief, and can relay the signal downstream very quickly so that your response can be quicker. So overexpression or increased expression of these cell surface receptors is the first subtype or the first strategy. Second, there can be some sort of an activating mutation. So compared to the normal protein, there will be a mutation within this particular protein, as a result of which it will stay active. Under this domain, there is, let me just mention the name and then one of you can just discuss your idea about it. Activating mutation is something that is supposed to be helpful for increased activation, right? It's a positive sort of, well, not feedback, but a positive direction for cell signaling. So if I say that a particular mutation in a particular receptor, say receptor X carries a mutation from C to T, from cysteine to threonine, and I say that this mutation is activating in nature, what does it mean? I would really like somebody else to try as well, but if Ankit, you know the answer, you please go ahead, or anybody else, any ideas you have, please interact. Let's have a discussion. I've not even heard your voices. So any idea, anybody?
No, ma'am.
Okay, anybody else wants to try? Harsha, Anshika, or Vaishnavi? Any ideas? Yes, I
No,
could hear you.
yeah, ma'am. No, ma'am.
You don't know? Okay. Um, okay. So basically, a constitutively active mutation, like I just mentioned, uh, say for example, this is look at this receptor itself. This receptor has a ligand bound to it. Say, for example, there is a mutation within this domain, this blue colored region, such that this protein is always phosphorylated. There is some change from, say, what, like I just mentioned, from cysteine to threonine, and that change allows phosphate group to always be bound here, or phosphate to always be present here. So then what happens? This receptor does not need ligand anymore. It can just work independent of whether there is a ligand or there is not, and as a result, that pathway will be constantly switched on. So this is a very dangerous situation, and it is something that happens very, very extensively in cancers. And as a result of that, what happens is that the cell becomes unresponsive to the ligand that is present. However, what is also lost is the negative feedback on it. So the normal route for negative feedback is that if this, for example, if this pathway is constantly on, there must be some mechanism within the cell which will try to shut down this pathway, and as a result, it will reduce the amount of the activator, which was the ligand. If there is reduced amount of activator, it does not make a difference because now this protein is independent. It does not care whether there's a ligand or not. This pathway will be constantly switched on. I think there was an image. Let me just back to it. In the previous class, I think we had just touched upon it. Yeah. So if you see this particular diagram, let me just zoom onto it a little bit. So this was an example where EGFR is also a cell surface receptor. So in this, it's not an RTK, but it's no, it is an RTK. It's a cell surface receptor. So what you see here is that no ligand is present on the outer domain. Nothing is bound to the protein. However, there are mutations within its intracellular domain, as a result of which this pathway is constantly switched on, and this is an RTK-driven oncogenic pathway. So basically, that is what I was trying to bring to your attention, that sometimes some pathways can be constitutively switched on, and these mutations, such as threonine to methionine or leucine to arginine, these are such examples of such mutations in EGFR that have been studied, and these can be found across multiple types of cancer. Were you able to understand it? Just unmute and somebody please let me know.
Yes, ma'am.
Okay, others? Hello? Am
Yes,
I
ma'am.
audible?
Yes, ma'am.
Understood?
Yes, ma'am.
Okay, good.
Yes, ma'am.
Okay, please let me know if you don't understand something. It's becoming a monologue otherwise. So if somebody contributes, that will be good. But it's okay. It's a lot of new information, so I understand you're just trying to absorb it. Okay, coming back here. If I go fast, please let me know. I'm more familiar with these topics, so it might happen that I'm just, rushing through something. But yeah, we don't need to hurry. We have a lot of classes. If we are not able to complete what I've decided for today by today, we'll discuss it in the next class. We'll keep the recap a little shorter then. So that is all up for, adjustment. Don't worry about it. So yeah, that's what I was mentioning. So let me just show this to you as well. Yeah. So see, like I just mentioned, EGFR is also an RTK. So we saw that there was a mutation in that example from threonine and methionine, that example that you saw. And if there's a constitutive activation of this pathway, so then the downstream RAS is on, the RAF is on, the MEK is on, and cell proliferation, cell survival, cell translation within the cell is constantly on and is at a very high level. So any control over its expression is lost. So in such cases, even treatment is quite difficult. There are ways to treat such patients, but it's just difficult, and the chance of responding to that treatment reduces. So yeah, that is one major way in which these RTK RAS signaling pathway dysregulation is a major challenge. And that is what is mentioned in the legend as well. So the first legend shows that overexpression, possibly due to gene amplification or due to altered transcription. Yeah, this is also another possible way that you have gene X. Either you have 50 copies of gene X back to back, or there can be that gene X is now under the control of some sort of transcription factor, which is itself behaving abnormally. As a result, it binds, and it binds with a lot of strength to the downstream gene. And as a result, there is excessive transcription of that particular gene, excessive translation, and therefore more expression of that gene. So both ways are possible. The second one, like I mentioned, is an activating mutation in either the extracellular domain or the kinase domain. As a result, the protein becomes ligand independent. So it is a constitutive activation of the receptor. The third example is of oncogenic fusions. So I'm not sure if you're familiar with it, but basically, have you heard about gene fusions? Are you familiar with that concept that there are fusions within chromosomes that happen during, well, sometimes it happens in at the time of development, like in little babies, they often have some very dangerous gene fusions. And of course, this happens a lot in cancer. Have you heard about it?
No, ma'am. I have not heard it.
Okay, then that's a lot of new information then. Basically, if anything is very unfamiliar for you, just note down the terminologies, drop me an email because I might forget by the end of this class, but you can note it down somewhere. And, we can just focus, give some time to understanding these terms because they are concepts that you learn when you learn cancer biology in more detail, but it might be unfamiliar for you. Just let me know. They're not difficult. You just need like a diagram to understand it clearly. So basically what happens is at the time of cell division, I hope you're all familiar with that process. So if you've read about the sub-steps of cell division, so you'll have all of the genetic material within the cell that will, from chromosomes, it will get completely, the organization will be completely lost and it will become genetic material. Sorry, the reverse. From the unorganized nucleus, you'll have the formation of chromosome at the time of cell division. And then the chromosomes will go through all of those different substages where it will be prophase, metaphase, anaphase, and telophase. So at the step of metaphase, if you are familiar with it, okay, are you people familiar with these substeps of cell division? If anybody is not, just let me know. We'll quickly look up an image on Google.
Yes, ma'am, we are familiar. Familiar.
Familiar? Okay. So during metaphase, if you remember that, think of a cell in this manner, and at the center of the cell, all of the chromosomes, they align, right? And then the two chromatids, which are like sort of like this, basically, the two chromatids, they separate in opposite directions, and they are divided equally among the two daughter cells. That is what happens. So this is the most ideal situation. That is what is expected. Now, in any cell, whether it is normal, whether it is cancerous, as even in normal cells, a lot of times what happens that chromosomes, they break. Why do they break? That is because during this process of alignment and during the process of separation, the more you read about it, it's an entire world by itself. It looks like the most obvious thing, that it's like a magnet, that this magnet is supposed to attach at the center of the chromosome and it's supposed to divide. That's not what happens. There are hundreds of signaling pathways that are operating to make sure that this happens correctly. And despite that, sometimes it happens incorrectly as well. So what happens is during these steps, sometimes chromosomal breakage occurs very, very frequently. Actually, it occurs. And if it happens, most of the time it gets repaired. If it doesn't get repaired, chromosomes break. Now, when they break, when I say chromosomes break, it means that the organization of the chromosome is lost. And after breakage, what happens is in most cases, you will not actually find like a broken chromosome just existing by itself, because there are enzymes within the cell which are known as nucleases, if you're familiar with it, which will digest that DNA, it will chew it up. So what is the tendency of such broken parts of chromosomes is to actually find something to attach to, something else that is also broken, two broken things, they attach. And that helps them to stabilize. What is this? This I'm talking about from the perspective of as if you are inside the cell. If you look at it externally, if you look at it from a scientist's point of view, what you will see are that there are gene fusions. Fusions means basically two genes which were not next to each other, they have suddenly come together and they have formed something which is known as a gene fusion. So now what will happen, whatever product you get as a result of this fusion might be, not always, but might be cancerous. Why so? Let me give you other examples. I thought you might be familiar with it, but there are certain very common chromosomal fusions as well. Like there are BCR-ABL fusions, etc. These have been studied for a very long time. But basically what people have understood is that in a lot of cases, fusions can be oncogenic because of the nature of the gene which has fused. Say, for example, um, say a gene X, which within a cell, a normal cell, under normal circumstances, say, I got locked out or something. Fine. Can you hear me? Can you see the screen?
Yes, ma'am.
Yeah. Okay. So a gene whose regulation is normally very tightly controlled, it might just happen that that control is suddenly lost. Why is it lost? Because it has now fused to something which is constitutively expressed. Say, for example, a gene which is very necessary for a cell to survive, that will be switched on under all circumstances. That is, say, gene A. And you have gene X, which is not supposed to be expressed. Now, there is a cut in both of these genes. Not a cut, but basically they break. And as a result of whatever circumstances or whatever, they fuse with each other. And now gene A and gene X are fused with each other. Now, the control at which gene X was normally used, which is to always be expressed, that control is now shifted on to gene X as well. So both the genes have started to express at very high levels. A, that is possible. They are expressed at very high levels. B, they're resulting in very abnormal proteins. Say, for example, what you see here, as you can see that there can be a fusion between the first part of the receptor with some other random part, or there can be fusion of this random part with the other part of the receptor as well. So what you're getting as a result is a very abnormal protein. So either one way it can be that this protein is just a non-functional protein, or it can be a hyperfunctional protein. Both things are possible. So in a lot of cases, these fusions are oncogenic, which means they are cancerous events. So more often, the kinase domain, it actually fuses to a different partner. And what it can eventually lead to, it can lead to oligomerization, it can lead to some altered subcellular localization, some very strange expression, strange stability, all of which leads to very abnormal activation of these receptors. So in this case, it might be that once this has bound and this receptor is switched on, the pathway is always on. Or in this case, you can see this also leads to a constitutively switched on pathway. So basically, things go wrong. That is the crux of the story. So oncogenic fusions are another very common subtype of events that very frequently occur. And finally, the fourth one is ligand dysregulation, which basically means that there can be either autocrine, or this can be either autocrine or paracrine in nature, which all results in receptor activation, and there can be various cellular responses to it, including proliferation and aberrant tumor growth. So ligand basically is functioning in an abnormal manner, as a result of which the pathway is functioning abnormally. So basically, these are the common ways. I hope you were able to understand it. I have introduced a couple of new concepts, so it may be confusing, but I hope you understood most of it. So overexpression, activating mutations, oncogenic fusions, ligand dysregulation, most common ways in which any signaling pathway can go wrong, but in context of RTK, this is very, very relevant because they are cell surface receptor proteins, and therefore they are receiving the signal from outside, and therefore their regulation is a lot more crucial. So yeah, anything you don't understand, you have to interrupt me. I can't even see your faces, so I can't figure out if you've understood it or not. But I really hope you do. And if you don't, I want you to communicate with me, please. This is another representation of these pathways. So RTKs is, I have just used multiple resources. Yeah, again, just to let everybody know, most of you weren't there in the first class. So I have cited the paper at the bottom. You don't have to read the paper if it is not something you feel comfortable with right now. You can just go look at these images, read the legend from there, the figure legend, and that will help you understand the concept that I'm trying to explain. You can look at videos, you can look at some study material, whatever helps you understand. It's about understanding. Use the internet to your best capacity. You'll learn a lot. So yeah. So different mechanisms by which RTKs are dysregulated that can occur by a gain of function mutation. Now, what is this? So mutations, you've all understood that there can be mutations which cause cancer. That is the basic story that even a layman knows in today's time. Now, what does this mutation exactly mean? So there can be majorly two types of mutations: gain of function, loss of function. As the name suggests, it is literally what the name suggests. So a protein which did not function in a particular manner has gained a function, as a result of which it is now oncogenic. Loss of function, a protein which was supposed to do its job in a certain way has lost its function because of the mutation. Simple meaning is that. So this is like when we talk about these mutations, most commonly they are grouped in these two ways because of the nature of the gene on which it has actually happened. So if a gene has gained this cancerous ability or the cancerous function, then it is in most cases an oncogene. So an oncogene usually has gain of function mutations, whereas a tumor suppressor gene, which is normally supposed to ensure that tumors do not develop, that has undergone a loss of function mutation. So it has lost its function and hence the cell now goes crazy and cancer is what happens eventually. So the first one you look at is gain of function mutations. Second, amplification. Like if you remember, we had just discussed it a while back. So you have multiple copies of the same protein. Chromosomal rearrangement, what I just explained as fusion. So you can see that the upper part of the receptor has fused with something strange pink. And here you can see the reverse has happened where the intracellular part of the protein has fused with that pink other protein, and that is also an abnormal fusion protein that has formed. Then you can have, this is something new, not something we've discussed till now, but there is tyrosine kinase domain duplication, whereby the domain itself gets duplicated and it has bound to something else. And so even without dimerization of the receptor itself. So there is no other receptor present. It's just that the domain has bound to itself. And that itself, if you remember, is the triggering event which will lead to phosphorylation and all the downstream activity. So that is also another way in which not mutation, but oncogenic activation can occur. And finally, the autocrine and paracrine activation. So basically what it means is that autocrine, as in, if you know the terms, autocrine means screen is usually used for, not screen, CRINE, crine or crine, as you, whatever you're comfortable with the pronunciation. So that is usually used for anything that is secreted. So for glands that secrete or that release hormones, you call them as endocrine or exocrine glands. So similarly, autocrine means something which is released by the cell or is secreted by the cell and which binds to the cell itself. So autocrine regulation is sort of like a positive feedback, can be a positive feedback mechanism, but basically a cell synthesizes protein X, releases protein X, and then protein X binds to the receptor present on the cell itself. That is autocrine activation. Paracrine, as in two cells which are adjacent to each other. Paracrine as in somewhere in the neighborhood. It's close to each other. They're close to each other. So paracrine activation, the protein synthesized by cell X, which you see here, this is cell X, is binding to the receptor present on cell Y, and that is leading to the paracrine activation. So in that case, this can also be a cancerous event depending upon how much protein is synthesized, what kind of receptor is it, what is it activating, and all of these downstream differences. These abnormal activation lead to all of these changes in cell proliferation, differentiation, angiogenesis, cell cycle, cell death, metabolism, basically the hallmarks of cancer like we have discussed. So all of this together are generating abnormal RTK activation, and they would eventually lead to the abnormal behavior of these cells. So this is how RTK or RAS signaling can play an oncogenic role. before we move to the next pathway. Any doubts, any queries, anything that I rushed by and you want clarified, please unmute and please tell me.
Ma'am, this dysregulation
Hmm.
mechanisms can also happen in other signaling pathways.
Yes, definitely. But some of them are actually unique to the RTK pathway. Like, for example, tyrosine kinase duplication. The domain duplication itself is good enough. When we talk about the steroid hormone pathway, there I'll show you that there is um, a sort of subtype of this exact process itself, whereby two receptors, again, we are talking about AR only. Those are the examples that come to mind because I work on it. So AR protein, it happens in such a way that the mutation that has occurred, these two proteins are bound to each other. The dimerization under normal circumstances is necessary to initiate that pathway. But because of whatever mutation has happened now, they are constantly bound to each other. As a result, that pathway is constantly switched on. So that is another way that is unique to steroid hormone receptor pathway. So some of them are actually unique to this pathway. Anything which you find is dependent on the fact that it has to be a cell surface protein. That is usually RTKs, because RTKs are cell surface receptors. Some of them can happen in GPCRs also, which are again also cell surface receptor proteins. But GPCR mutation has a lot of other different effects also. We'll not go into it right now. But yes, some amplification or rearrangement, all of these can happen in any pathway, and it is going to have very horrible, very drastic effects.
Ma'am, another question, like in different types of cancers, different signaling pathway dysregulation mechanisms could happen, right? Like
Hmm.
in some cancers, there must be different signaling pathway getting dysregulated, and in some other cancers, it's different.
Hmm.
Is it possible?
Yeah, that is actually what happens also. So, um, just give me a second. I am not sure if I included that slide, but if not, I'll show you the paper. Give me a second. I don't think I have it in my slide. So let me just show it to you. This is the paper I was discussing. So, mm, yeah, I don't know if, don't be scared by the diagram if you don't know how to interpret these right now. It's fine. It's something you learn in your PhD. You don't have to completely understand it right now. But basically, see, here you have what you see at the bottom are different types of cancers. Okay, like GBM is for glioblastoma. Um, uh, Let me take any other example. This is for pancreatic adenocarcinoma. This is PRAD is for prostate adenocarcinoma. All of these. So these are different types of cancers. What you see at the top on the left are different types of proteins, different types of RTKs and other proteins, of which what you see in red is an oncogene. What you see in blue here are tumor suppressors. And what they have shown overall are the number of patients that carry these different types of mutations. So you can see that, for example, EGFR, from the total number of patients that they had studied, 50 of them carry a mutation, 50 of these samples carry a mutation in the EGFR protein. What type of mutation is it? Here in red, it is an amplification mutation, which means that EGFR was present in more number of copies. However, you can see that the frequency at which it occurs in different types of cancers is very different, but it is present. Again, look at any other example, take ERBB2. ERBB2 has different frequencies. You can take any example like this. So there are some, like if I have to give it a more direct answer, basically any mutation can occur in any pathway. You cannot, and it's not even unique, multiple mutations, multiple types of mutations. Like right now, it might appear to you that in any cancer, in a particular patient, only one type of mutation has occurred. No, same patient can have same, a gain of function mutation also, an amplification also, mutation in three different pathways also, lots of things can happen. It depends upon the nature of the disease. But what is usually understood is that certain changes or certain alterations are more common in a particular type of cancer. That's all. That's just the more easier way to diagnose a disease. What can happen is limitless, but what is the most common way is what is used for diagnosis. Like I gave the example in the last class that if there is a mutation in BRCA1, BRCA2 in a particular patient's family, they have a higher propensity of developing breast cancer. But does that mean that if they get a mastectomy done, there is zero chance of developing cancer ever again? Well, honestly, no. They can develop other types of cancers as well, but they have the highest propensity of developing cancer of the breast. But yes, it's possible that they can, because BRCA mutations can also be present in other cells of the body, and it's a necessary protein that is part of a repair pathway within the system. So if a repair system is shut down in some other tissue of the body, there can be cancer of that tissue as well. So it's about a chance event where it is occurring and whether or not that event alone is capable of becoming a cancerous event or not. These are all chance events. What we are trying to understand are just pathways right now, which can individually or in combination go wrong within a patient. Was that okay? Did you understand?
Yes, ma'am.
Yeah, this also reminds me one more concept I would want you to understand is that there are two types of events. I should have put it in the slide, but if you're making notes or something, or just if you can recall, like remember it better, two types of events which are called as driver events or driver mutations, and then there are also passenger mutations. As the name suggests, it's very blunt. Basically, any event which is driving or which is sufficient to cause cancer is a driver event. Any mutation that is present along with a driver mutation and that helps in the development of cancer but is not capable to cause cancer by itself is a passenger event. I hope you understood it. So a driver event is something that is oncogenic by itself. Say, for example, like you saw EGFR mutations present in 50 patients. It is known to be a driver event that if there is a mutation right at the top of this cascade, it is going to completely go haywire. But along with EGFR, if there are mutations in, say, 5, 10 other pathways also, then that is going to help the mutation become much worse. When I say worse, it means from a healthy perspective, but from the point of view of cancers, it is going to make the cancer a lot stronger and the disease way worse. So these are two more terminologies that you should learn. You'll understand it eventually. Right now, what we are discussing are majorly driver events. These are more literature based. How do you segregate the two? Well, very difficult to explain right now. Um, you can do it in the lab. Basically, you can take if you've heard about cell lines or worked with cell lines are basically patient-derived human patients or human or mouse or whatever, which type of model you're using depends on that. But mostly if you're talking about human cancers, you will take human cell lines. So you will have to take a normal cell line, which is representative of a normal human being. And within that cell line, you will have to either increase the expression of that gene or decrease the expression of that gene depending upon what is the function of the gene itself. Say, for example, if I take a um, normal cell line for that particular type of cancer and I overexpress EGFR in it, if I see that that cell is becoming cancerous, then that means it is a driver event. If it is by itself not able to cause cancer, but I need to have 3, 4 mutations in parallel with it, then it is possibly a passenger mutation. So these are different ways of studied. Again, this might be too advanced or complicated. Let's leave it aside for now. Come back to the slide. I am pretty sure I'm not going to be able to complete today, so don't worry. We'll just discuss the PI3K pathway quickly. Um, it is very similar to what we have already discussed, but yeah, you should definitely learn about it. So PI3K is a kinase basically. Read about its full form. I have not put it anywhere in the slide, but the reason why I discussed it right after the RAS cascade, the RTK RAS signaling cascade, is because this is also part of the RAS signaling pathway, or it comes downstream of the RAS signaling pathway, and it also definitely also has an oncogenic role. So let's go over it. So what do you see here? Just if you give a overall look at the pathway by itself, this is the RTK, like we have already discussed, the receptor tyrosine kinase, to which the ligand is binding. Ligand can be of different types, like insulin, like growth factor, cytokines. Growth factor example being the one we have been talking about, EGFR, epidermal growth factor receptor. That is a receptor, but its ligand is EGF, which is epidermal growth factor. Or it can be some sort of cytokine. If you're not familiar with cytokines, think of it as a chemical within the cell, which is which has a lot of role to play in immunology, for immune cell or immunity regulation. And you definitely must have heard about it during COVID, where everybody was talking about cytokine storm and all of that. But yeah, don't just go by hearsay. If it's interesting to you, read about it. It's just a way in which cells talk to each other. It's a communication molecule. So yeah, so what you see here is that receptor tyrosine kinase is getting activated because of this particular ligand binding. And I'm also reading the legend along with it. And there is phosphorylation of the tyrosine residue of its cytosolic domain, like we have already understood. What does that do? It creates some scaffolding region, which allows the recruitment of the lipid kinase PI3K. I put it here. It was not there in the first slide, but anyway, PI3K or phosphinoinositide 3 kinase to the plasma membrane. Lipid kinase, as in it phosphorylates lipids. So this is PI3K. The activation of the RTK allows PI3K to be recruited onto the cell membrane. PI3K then phosphorylates something which is downstream of it, which is known as PIP2. Not something you need to worry about. This is just a long way of writing PIP2. PIP2 means it has two phosphates. When PI3 kinase will act upon it, another phosphate would be added and then it will be called PIP3. And also it phosphorylates something else known as P10, which is a lipid phosphatase. And this PIP3 acts as a second messenger, which recruits the serine threonine protein kinase, AKT, to the plasma membrane, where it gets activated by phosphorylation at threonine 30H and serine 473 by PDPK1 and a mechanistic target of mTOR protein kinases, respectively. Eventually, AKT signaling serves to promote cell survival, cell growth, or cell proliferation by various changes to cell metabolism. What you see in hexagon are common points of activation and inactivation by cancer-associated mutations, and P indicates phosphorylation events. Okay, lots of jargon, lots of confusion. Let's streamline it. This part you must be familiar with already by now, right? RTK is present at the cell surface, ligand binds, RTK is activated. RTK allows the docking of some other proteins like adapters, and it also allows the activation of PI3K, which is a lipid kinase molecule. What does it do? Lipid kinase moves towards the cell membrane, and because it is a kinase and because it is a lipid kinase, it will add a phosphate group onto a lipid. Which lipid? PIP2. The full form is, it's too long. It's basically an inositol molecule. I don't know if you guys have studied it, which is why I did not want to go into details. Just any lipid X. It's an inositol, a type of lipid onto which two phosphate groups are present at position 4 and position 5. 4 and 5 are representative of the carbon at which phosphorylation has occurred. So PIP2 gets phosphorylated and forms PIP3. Why is this important? That is because PIP3, otherwise also known as IP3, is a second messenger within the cell. What is a second messenger? So within cells, there are certain molecules that are called as second messenger, which goes and broadcasts information within the entire cell. Think of it in this way. You have an external stimulus, which is binding to the RTK, and something which is coming in between. This is a primary messenger, or this adapter may be a primary messenger, which is carrying the information which has come from outside to one or two other molecules inside the cell. Secondary messenger will carry that information and just spread that information everywhere. What does that mean? It means that this messenger is then responsible for either activating or inactivating multiple pathways downstream of it. One example of this is IP3 or PIP3. Another example of this is calcium. We'll come to calcium when we if we discuss GPCR in the next class. So calcium also is a second messenger which goes and then activates several different pathways. It does not know how to keep a secret. It goes and broadcasts information everywhere. This is one thing that happens. Additionally, what PI3K also does is that it creates sorry, this is where I was, yeah. PIP PIP2 to PIP3 is carried out or is executed by PI3 kinase, the enzyme that we are talking about. This acts as a second messenger, and what does it do? Yeah, just a second, Ankit, I'll come to you. And then it recruits AKT onto the cell surface, and then AKT is responsible for several growth and metabolic changes within the cell. The dephosphorylation of this is carried out. What is the dephosphorylation? Removing that phosphate group. That is carried out by P10. And P10 is extremely crucial, and the mutations of P10 are something that are very common in a lot of different types of cancers, especially the one I work on, again, prostate cancer. P10 mutation actually leads to spontaneous development of cancer, or at least precancerous stages. So that is the overall pathway. I'm sure it is very confusing. Please go ahead with your questions.
Ma'am, this phosphatidylinositol, the PIP2 and PIP3 are embedded in the plasma membrane, or is it a part of plasma membrane?
Here it can be a membrane embedded structure, PIP2. It depends upon no, in this case, it is actually membrane embedded only because a free lipid cannot exist within a cell, right? Either it is part of the plasma membrane, or it can be part of some, like, how do I explain? Have you studied about lipid drafts, which is like a small, think of it like a little boat made up of lipids. So it diffuses from the cell. It can become part of a vesicle. It can, move to other parts of the cell, but it cannot remain just floating within the cytoplasm because it is a lipid, and the interior of the cytoplasm is not going to allow it to survive, right? It is aqueous in nature. So it is part of the membrane, but when I say it's a secondary messenger, or it's a broadcaster of information, that is because the PIP3 will then allow the docking of lots of different things. One example of which is what you're seeing here, that AKT now binds to this PIP3, and AKT is then able to execute all of the downstream things. Similar for P10, like you see, it is in this membrane bound format. Not format, it is part of the lipid membrane, and then it gets dephosphorylated and it remains a part of the membrane. What I said, may or may not be a part of the membrane, was for calcium, because calcium release actually occurs downstream of it, and it is not a membrane bound structure. It is something which gets released into the aqueous part of the cell, and then it moves around and does whatever it has to do. But this only remains membrane bound. I think I'll show you this pathway. If you had seen this slide, I think this one. Yeah, if you had seen this slide, here also the same thing had been shown. I just did not go into details about it. But after this RTK activation, PI3K is getting phosphorylated, or PI3K is becoming active. It is converting PIP2 into PIP3. And then PIP3 is activating AKT, which is phosphorylated, and then through some different complexes. The main target of which, what we are very interested in, because it's a huge uh, I'm not sure if it is very prevalent today, but yeah, at one point of time, mTOR was an excellent target for cancer therapy. Literally, mTOR stands for mammalian target of rapamycin. Rapamycin is a drug which is used for different types of purposes, but in this case, rapamycin is not an anti-cancer drug. But mTOR and mTOR regulated pathways have been of huge interest in context of cancer. So yeah, coming back here, I hope I answered your question. But then, yeah, just logically going by the definition of lipids, if you have studied how micelles form, and if you have I'm sure you've studied it as part of chemistry at some point of time, that a lipid always has to either, form like a lipid bilayer, or if it is a monolayer, it will form a micelle. It cannot just exist in free form. So here it exists as part of the bilayer, but it acts as a secondary messenger. Uh, yeah. Now, yeah, apart from that, the rest of it, did you understand? I just moved ahead, sorry. Anybody, any other questions? I might be going fast. Please let me know. We'll wrap up in 7-8 minutes. Don't worry. I'll only discuss the AKT pathway for today. So just any doubts, please let me know. Okay. If not, then what happens after AKT is activated? We already discussed the fact that, it helps in cell survival, all of those other pathways. But also AKT regulates cellular metabolism through several downstream substrates. For example, TSC2, it has a component of the TSC complex which activates mTORC. All of these are downstream pathways. You don't need to at all put a lot of effort into remembering it. But basically understand that AKT, downstream of AKT, you have mTORC1 and you have a lot of different types of regulators which have huge roles to play in cancer. One major component is MYC, MYC, MYC. It's a very dangerous protein. Let me just put it this way. It is an oncogene. It has immense roles to play in most kinds of drug resistance that you will hear about, that more often than not, most drugs, they try to shut down some pathway or the other, and by some other bypass pathway, MYC starts overexpressing and cancer cells become resistant, and all that effort you put into developing that drug goes to waste. So it's a very dangerous protein, but a very smart protein. So it's wonderful to study about, but very complicated when it comes to applying any technique or any sanctioned therapeutic strategy. But then basically, I'm not going into details about it. It's a lot of jargon. You can just read about it. That is what I would suggest. But basically, at the top of the entire mountain, if you think about it like a mountain, at the top you have you have your RTK. Downstream of it is PI3K, phosphorylates PIP2 into PIP3. PIP3 is now a secondary messenger, activates AKT. AKT is able to regulate cell survival, growth, proliferation, and cause many metabolic changes. It is aided, or this phosphorylation of AKT is aided by mTORC2, and dephosphorylation of the PIP3 into PIP2 is carried by P10. Therefore, it acts as a tumor suppressor, and therefore it's a very important thing to have functional within the cell under normal circumstances. Also, this is actually a better example of understanding how yeah. Yeah. How AKT is very important in this pathway. It regulates metabolism through a number of different downstream transcription factors, including FOXO, HIF1, MYC, like I already said, ATF4, SRABP, NRF2. All of these are necessary, and they are controlling some very important pathways. For example, glucose uptake, glycolysis, synthesis of nuclear, all these synthetic pathways for that matter. If you have studied metabolism in your undergraduate or something, you would be very familiar with AKT by now, because it sits at the top and it just controls a lot of things inside the cell. Lipid synthesis, pentose phosphate pathway, PPP, redox balance, all these metabolic pathways are under its control. So uh, if within a cancer cell, there is some sort of activation within this pathway, which leads to continuous switching on of AKT, then what will it do? It will synthesize all these things that a cancer cell needs. So you remember how I said that there's a crunch for resources for a normal cell, so it is very smart and it utilizes everything optimally. For a cancer cell, it doesn't bother. It uses all the resources. It synthesizes its own resources. It creates ways of surviving. It uses host resources for its own needs, and it's just brilliant. But yeah, it's able to do all of these things because it switches on all these kinds of pathways. So I think we'll stop it here. We'll come to TP53 signaling pathway in the next class. I'll go to the Yeah, maybe I'll go to the last concluding slide for today. We had a little bit left, but it's okay. We can wrap it up in the next class. But what are the key takeaways for today? First of all, several signaling cascades work in concordance with each other to maintain the physiological homeostasis of any normal cell. The negative regulation of such cascades is very essential to maintain normal cell proliferation and maintenance. Dysregulation and imbalance in the functioning of these pathways is indeed a key oncogenic event in various cancers, and they are very potent therapeutic targets for the clinical management of cancer patients. I removed the questions because it's better to answer them after we have discussed the rest of the things that were meant to be completed in this class. But don't worry. Any questions? I introduced a lot of new things, I know, but any questions, any queries, please go ahead. Anybody? Did you understand? Is there any feedback from anybody? Do you want questions? Do you want more time to be spent on any topic? Anything that you feel is necessary? Please go ahead. I would appreciate any feedback. Yeah, Harsha, go ahead.
Ma'am,
Yeah, anybody? Yeah, one of you go ahead.
ma'am, if possible, could you please explain in the next class the basics of this signaling, please? Like how
Hello.
when the receptor binds to the ligand and
Mhm.
how they activate the other proteins, like how the secondary messenger activates the other.
Acha. Okay. Okay, yeah, sure. We can keep like a few slides. Yeah, it won't take long. I'll explain it. Sure. Yeah, Harsha, go ahead.
Ma'am, actually, can you share the papers which you have cited, ma'am, for that hallmark part?
I have shared the PPT. Did you not receive it?
No, ma'am. Yes, ma'am. I go through it, ma'am. Ma'am, actually, last class you told that three questions to discuss, ma'am. Three, this one, that hallmark one and auto plus this one.
Yeah. Yeah, so I did
Yeah.
not discuss it today. You want it to
Yes,
be
ma'am.
discussed, then we can. Okay, fine. Maybe we'll discuss it in the next class. So I'll keep these
Okay.
two things. We'll split signaling for a few more classes because I can feel it is getting a little heavy. So we'll split it and we'll discuss the questions. Were you able to get answers for it?
Yes, ma'am. I brought, but I couldn't speak, ma'am, because of network coverage.
Okay, no problem. I can hear you now, but yeah, you were not very audible previously. But okay, we can discuss it. No problem.
Okay. Thank you. Yeah.
Okay. Thank you for joining and have a good night. We'll meet again on Thursday. Please try to be on time because we're keeping it a little crisp. But yeah, it's okay. Don't worry. And any questions, drop me an email. I'll share whatever is needed for that. Okay. Thank you. I'll end the class now. Good night.
Thank you, ma'am.